Hello, people. Celine Ramos, gallery view. So it looks like we got 7 people, a bunch of people missing today. I'm trying to figure out, all I can see is my own face. It's like big ugly guy right in the front here. How do I see everybody else? Mike West is up. Yay, Mike. So can you guys show your faces real quick? There's Bizwit, Celine Ramos, Adriana, Ariana Totaram, still one of my favorite names. Is that it? 1, 2, 3, 4, 5, 6. It's 6 out of 8. That's not so bad. 6 out of 7. 9:30. I'm going to wait a little bit because I don't have all that much today. We're going to be talking about the contraction of the cardiac cell and how that's regulated by cyclic AMP and beta adrenergic agonists. So I think it's only going to take me 45 minutes or so. And then I'm going to talk to you about a paper I want you to read for Thursday, because we'll sit and talk about this paper. So I like to note, by the way, when you talk about diversity in Plattsburgh State, look around this screen. There's only 2 white guys here. The rest of you people are people of color. That's awesome. That's really cool. One of the strengths of Plattsburgh. Oh. Shall we go ahead and get started? Someone just came. All right, let's go ahead and get started. I don't have to take up your whole morning here. Let me see if I've got my PowerPoint ready. Hang on, I don't think my PowerPoint's open. Just one second. Okay. And now we shall share. Sharon is Karen. Karen is Sharon. There we are. Okay. So this picture is kind of what we're talking about today, but it's just something I grabbed free off the internet to kind of show up the front page. Okay. So we're going to be talking about cardiac cells. Now, cardiac cells, I don't know, I think a lot of you have had me for cell biology, and I talked mostly about skeletal muscle cells there. Skeletal muscle cells go from one end of the muscle to the other, and they're multinucleated because a whole lot of small cells fuse together to make one big, long, multinucleated cell. Cardiac is like as if those cells never fused. So there's all these cells that are lined up end to end to end to end, but they don't fuse together to make one big cell. They're still individual cells. So each of these little square boxes here is a cell, and this stitching here are intercalated discs and gap junctions between the cells that hold those cells together. So you get a lot of, so you have strength in this direction, right? Because it's going to contract and it needs to pull on each other. So each cell has got like 1 or sometimes 2 nuclei, usually 1. So these are small kind of, I like to say square, because if you're looking at them in 2 dimensions, they look square. Actually, they're cylindrical. I used to do work with these. I'm going to BS a bit here because I've got time to kill. I used to do work with these when I was in grad school. What I did was I took a hamster hearts, hamster for some reason, I don't know why, took hamster hearts, pulled the heart out, and then you put a cannula in the aorta, and then force fluid backwards through the aorta down into the coronary arteries. So you perfuse the heart. So you can keep a heart alive like that, especially a rat heart, like for a day probably. They stay alive a long time. And they just keep beating because they're automatic. And we would take that and we'd circulate collagenase through it. So collagen is a lot of the protein that holds these cells together. So we dissolve that collagenase, dissolve the intercalated discs, or the connections anyway between the cells with collagenase, and then take the heart down and kind of shake it, and the cells would all fall apart. And you take those and actually do experiments with individual cells. We were measuring the contraction of individual cells. That was pretty cool. Didn't get a paper out of it, but it was fun to do anyway. Okay, so those are our cardiac cells, kind of cylindrical, square on either end. They tend to branch like this. There's a lot of branching that happens in these cells. So here's kind of individual cells here. These are not striations. Those are the intercalated discs between cells. And here's what that looks like. So this is kind of important. Here's your disc, the desmosome. That's what's holding the cells together. But also you've got these things, which are these are the gap junctions. So these are channels that go from one cell to another. So you've got a channel here and channel here. They touch in the middle, and there's cytoplasm can go back and forth between the two. But mainly what's important about this, less so than the cytoplasm, is the fact that these cells are now electrically connected. So we call it an electrical syncytium because it acts all as one. When one part of the muscle gets stimulated to get an action potential, that will spread all over the whole the whole cardiac muscle. And that's how you get contraction of the muscle to happen all at once. So all the cells are contracting at once to get that blood out of the heart. So that's kind of important, our gap junctions. Now, when we talk about the action potential, we talked about this, again, some of you have had my classes before, and I talk about the action potential in nerves in the drugs class, the illicit drugs class. And this action potential is different in cardiac because in the drugs class, in the neurons, I mean, you get sodium channels depolarizing, then potassium channels come on and it repolarizes and goes into an overshoot, and then those channels close and it levels off. So very fast. This action potential is very different in that you've got this plateau phase here. So a skeletal muscle or a nerve action potential will be about 5 milliseconds total. This is about 500 milliseconds because here in the middle, you've got two things happening. So, okay, so your sodium channels open here, you get depolarization, your sodium channels inactivate, and slightly behind that inactivation of the sodium channels, you get opening of the voltage-dependent calcium channels. So these VDCCs open here, and at the same time they're opening, you've got voltage-dependent potassium channels opening. So the potassium channel is potassium going out, that would repolarize. The calcium channel is calcium coming in, positive charges in, negative positive charges out, so they're kind of balancing each other. Because of this slope, you can tell that the potassium's winning, right? If it was just calcium, it would stay up here, but it's coming down, 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 down. So potassium's winning a bit, but you've got calcium and potassium, calcium in, potassium out, and that's what gives you this plateau phase. And then here at 4, the calcium channels, the VDCCs will inactivate like the sodium channels did. So these guys inactivated really fast. These activate a lot slower. Once they do inactivate, you've got only potassium current here. So potassium repolarizes the cell, and then those potassium channels close and you go back to rest, okay? So the key thing about this action potential that you should know is this plateau phase, because that represents the calcium coming in. And that's gonna be key to our contraction, is that calcium. Okay. Okay, so this is the sequence of events that causes contraction. I have to go slower. We're going to be done in 10 minutes here. And this is a lot of material. It's just that it's fast when I'm throwing it up on PowerPoints real quick like this. Okay, so the cell is stimulated electrically. How does that happen? We'll talk about that in probably about a month when we talk about contraction of the whole heart. But here we're talking about the ventricles. So basically, each cell is going to be stimulated either by the cell next to it or by the Purkinje fibers that we'll talk about, which are kind of nerve-like muscle cells that bring stimulation to the cells. So anyway, just accept the fact, because I'm telling you, and I would never lie, accept the fact that the muscle cell gets stimulated, gets depolarized a little bit. When that happens, it throws an action potential and the VDCCs open, and that allows some calcium in. That's our trigger calcium. So that's not enough calcium to cause contraction of the cell, but what that calcium is going to do is bind to the ryanodine receptors. And the ryanodine receptors are large channels on the sarcoplasmic reticulum. So in muscle cells, we call the smooth endoplasmic reticulum a sarcoplasmic reticulum because it's really modified and specific for muscle cells. So you've got this sarcoplasmic reticulum, SR, which is filled with calcium. Calcium's being pumped into the SR by the circa, we'll talk about in a second. And so it's really full of calcium. And so the trigger calcium comes in, hits those ryanodine receptors, they open, and calcium floods out. That calcium then increases calcium in the cytoplasm, and that does the same thing it does in skeletal muscle, which is it binds to the troponin C of the troponin complex. Troponin complex are 3 proteins that bind to the actin and to the tropomyosin. So let me hang on a second. I think I'm going a little fast here. Nope. All right, back up. So let me back up and talk about the contractile apparatus real quick because of this stuff here. So when the skeletal or cardiac muscle is relaxed, you've got thick filaments and thin filaments, just like in skeletal. And tropomyosin is covering the myosin binding sites on the thin filaments. So myosin can't interact, and that allows the cell to relax. And so tropomyosin is covering those sites, and troponin complex, 3 proteins, are controlling the position of that tropomyosin. So when there's no calcium, tropomyosin is blocking those binding sites. When calcium comes, it binds to troponin C, that pulls the tropomyosin complex, and that pulls the troponin complex, that pulls tropomyosin off the myosin binding sites, and then you get cross-bridge cycling, and the muscle contracts. A lot faster than this, but kind of like that. Okay, so that's what's happening here. Your trigger calcium binds to the ryanodine receptors. They open, cytoplasm increases in calcium really fast. Calcium binds to troponin C, changes conformation of the troponin complex. Troponin pulls tropomyosin out of the way, and then actin and myosin cross-bridge cycling happens and the cell contracts. Okay, let's open this up to the crowd. Quick question, because I know you know this, or you should know this. Who knows the 3 different proteins in the troponin complex? I talked about troponin T, C. Oh, shoot, I just gave you another one. Okay, troponin C, troponin T, and what's the third one? For a buck, I'll mail you a buck. You know that's a lie. Troponin T, troponin C, and troponin... I can't tell you this. I don't really know. What's Dante say? Troponin I, exactly. Okay, troponin I, which is inhibitory, is why they call it I. Okay, so let's go back to our share, because troponin I is going to be important shortly. Okay. Okay. All right, so then for relaxation, for the second half of the cardiac cycle, remember this thing's kind of constantly contracting and relaxing, so both are important. So your ryanodine receptors close, calcium stops leaving the SR, and this is closing because the action potential is over, there's no more trigger calcium. And so then you've got circa, which is your sarcoplasmic reticulum calcium ATPase, is going to pump calcium into the SR. Your sarcolemmal calcium ATPase, which is a calcium ATPase on the sarcolemma. Sarcolemma means the plasma membrane of the muscle cell, sarcolemma. Get to know that word because they use that a lot in cardiac stuff, sarcolemma. So you've got a calcium ATPase on the plasma membrane, sarcolemmal calcium ATPase, and you've got this guy, Mr. Sodium Calcium Exchange. We talked about this in cell biology. It's really important in cardiac cells. So this is also helping to remove calcium. So the sodium calcium exchanger then is indirect active transport. You've got sodium coming in for calcium going out. So that's going to help get some calcium out as well. And we'll see how regulation of that can regulate the contraction as well. Okay, so when this all happens, you're pumping the calcium into the SR and then out of the cell with these two. When that happens, calcium cones off Mr. Spelling. Comes off the troponin C. Troponin complex goes back to the normal rest state. Tropomyosin covers the myosin binding sites, and that stops cross-bridge cycling. And if you're not cycling, you're relaxing, okay? So basically, calcium decreases in the cell, tropomyosin goes back to its normal spot, and then the muscle relaxes. So let's go through that again while looking at the figure here. So here is a cardiac cell. This represents a transverse tubule, T-tubule. We saw them in skeletal muscle. Again, I keep assuming you all had me for cell biology. I think most of you did. This is a transverse tubule, which is an extension of the plasma membrane that comes down into the cell. So it's kind of like you've got the cell, but you've got fingers of plasma membrane shooting down into the cell. In this T-tubule membrane, you've got a lot of voltage-dependent calcium channels. And I don't know if you remember, but in skeletal muscle, these were directly touching these ryanodine receptors. Here, they're acting as calcium channels, so they don't touch the receptors. But when the action potential comes down this T-tubule, they open and calcium comes in. So that's our trigger calcium. So that's the calcium that's causing that plateau in the action potential, is that calcium coming in, which is really cool. By doing electrophysiology, you can like directly watch calcium coming into the cell. So anyway, that's the trigger calcium coming in. That comes in, hits these ryanodine receptors. Calcium floods out into the cell. It hits the contractile apparatus, troponin C, troponin complex moves, tropomyosin moves, cross-bridge cycling, contraction. Okay. Relaxation then, Relaxation then happens when your SERCA puts calcium back into the SR. Sarcolemmal calcium ATPase pumps it out of the cell, and your NCX over here is going to exchange calcium for sodium, and then calcium drops. Now, there's a couple other proteins here I'm going to throw out. Didn't talk about them in cell biology. They're going to be important here, though. One is FKBP 12.6. Don't you love it when they name things like this? I don't know what FK is, but BP is binding protein. So it's a binding protein that binds to FKB, and it's probably a molecular weight of 12.6 kilodaltons. That's usually how they name them like this, but I wish they'd give it a name. But anyway, FKB 12.6 is a small protein that binds to the ryanodine receptor, and the language they use is it stabilizes the receptor. Basically, what it's doing is decreasing the amount of calcium that comes out of it. So FKBP inhibits the ryanodine receptor. Now, it's not like a total inhibition where no calcium will come out. It just kind of modulates it. So when FKBP is bound to the ryanodine receptor, less calcium comes out. Over here, this purple guy here is phospholamban. Phospholamban is really important. So phospholamban is similar to FKBP 12.6 in that it binds to the SERCA and inhibits SERCA. So it decreases the activity of the calcium ATPase. So under normal conditions, non-stimulated conditions, your SERCA is being inhibited and your ryanodine receptor is being inhibited. And so and why is that? Oh, let's throw that one out to the crowd, as long as we've got time to talk. Well, why do you think that would be? Why under normal conditions would you inhibit the calcium, the SERCA, and the ryanodine receptor? For $10, $100, what the hell, I'm not going to pay anyway, for $100.
So that the heart doesn't misfire by other sorts of calcium coming into the cell for other reasons?
That's an interesting.
It has to be so unified. I'm just kidding.
That's not what I would say, but that's entirely possible, that it's kind of protecting the cell from extraneous contraction activity or something. It's not what I would say, but that may be part of it. Anybody else? What happens when you exercise? What happens to your heart? It
Beats
just
harder.
It's the same, right?
Same rate. No. It goes up and down, right? It gets faster and harder. I know that, but
it goes faster and stronger. So you're inhibiting these proteins because that gives you some room to grow, right? If you stop inhibiting these proteins, you can get a lot stronger. So that's what these things are all about. They're like putting gain in the system is what we say. In other words, you've got the ability to make this stronger. Okay, and that's our next topic. How do we make the contraction stronger? So anything else to talk about here? No. All right, so the next thing to talk about then is when you do exercise, you're going to need more oxygen to your tissues. So you need to increase cardiac output. Cardiac output, I'll talk about later today, is basically a combination of how fast your heart is beating and how strong it's beating. I got to check on my dog. Hang on. Okay. So this, when you stimulate a ventricular cell with beta agonist, it's going to beat stronger. You're going to get a stronger contraction out of it. So this is unstimulated. This is a normal contraction. I don't know. I made the curve up. It may be shaped a little differently. It's been a while since I looked at it, but just assume this is a normal contraction and this is a stimulated contraction. It's a lot stronger and it's a lot shorter in time, right? So you're going faster and stronger. The stronger, okay, means that however much your heart fills, you're going to inject more of that blood. So your stroke volume is going to increase. We'll talk about stroke volume later, but basically you're getting more blood out of the heart, and so there's more blood going into your system. And shorter contraction because beats are coming faster. So the heart has to get ready for the next beat. So it's going to go stronger and shorter. Okay, so what happens is in your cardiac cells, you've got beta-1 subtype of the beta-adrenergic receptors. The beta receptors, at least in this class, we'll talk about beta-1 and beta-2. Beta-1s are in the heart, beta-2 are in the respiratory system, your bronchioles and things, or what do you call them? You know, when you use your albuterol, when you've got asthma and you use your albuterol, albuterol is a beta-2 agonist, so it relaxes your, Mike's got it too. It relaxes your bronchioles. So beta-2 is in the bronchioles, beta-1 is in the heart. So that's why they use beta albuterol for your lungs, because you'll absorb some of it, but it's not going to affect your heart. It's not going to make your heart race. So anyway, cardiac cells are beta-1, which I think the drug is dobutamine, if I'm not wrong. We'll talk about drugs later. Okay, so when your beta-1 agonist or beta-1 receptors are activated, they're either going to be activated by epinephrine if you've got, a large stressful thing going on. You've got epinephrine release. That's a hormone, comes from your adrenal medulla. Or norepinephrine just comes from your sympathetic nervous system. So you've got sympathetic innervation of the heart, and norepinephrine is going to stimulate those beta-1 receptors. When you activate the beta-1 receptors, you activate GS, which activates adenyl cyclase, produces cyclic AMP, all that stuff we talked about in the last class. Cyclic AMP increases, and in the last class we said it activates protein kinase A, and protein kinase A phosphorylates targets. So we were very vague about it then. Whenever I speak about it in classes, I'm vague because the targets depend on the cell. So today we'll talk about the targets in heart that actually make the heart beat the way it does when it's stimulated. Okay. You've got targets that are going to make the heart beat stronger and targets that are going to make the heart beat shorter, okay? And both of these things are important. So first off, increasing the strength of the contraction. Here's the key. The most important thing is this beta stimulation, cyclic AMP, protein kinase A is going to phosphorylate your voltage-dependent calcium channels. That will increase the trigger calcium. Increasing trigger calcium increases activator calcium, and you get a stronger contraction. So that's the most important thing that's going on here, your voltage-dependent calcium channels. Number 2, phospholamban. So remember, phospholamban was inhibiting the SERCA. You phosphorylate it, it comes off the SERCA, and you get disinhibition of SERCA. That means SERCA is pumping calcium stronger into the SR. So the SR gets loaded with more calcium. And so now you've got more calcium in the SR, and when the activator calcium comes out, there's more activator calcium. So you get a stronger contraction for that reason. And our third target then is FKBP 12.6. Again, just like phospholamban, you're going to phosphorylate it. It'll come off the ryanodine receptor, and you get more activator calcium because more comes out the ryanodine receptor. So you're getting a stronger contraction here with beta stimulation because you're getting more trigger calcium coming in, which is going to produce more activator calcium. You're disinhibiting the SERCA, which means you've got more calcium in the SR, which increases activator calcium. And you are phosphorylating FKB 12.6, which disinhibits the ryanodine receptor, which gives you more activator calcium. Note, it's all about the calcium, right? It's all about getting more calcium out there. Okay. Now, to make the contraction shorter in time, we're going to have to relax faster. Here's how we do that. So phospholamban, again, you're disinhibiting SERCA, increasing calcium pumping, so the cellular calcium decreases faster. So phosphorylating phospholamban does two important things. It loads the calcium, the SR more, so you get a stronger contraction, and it loads it faster so that you get a faster relaxation. Now, troponin I talked about troponin I before. So when you phosphorylate troponin I, that decreases the sensitivity of troponin C for calcium. So that means it takes more calcium to get troponin C to pull tropomyosin off the myosin binding sites. So that allows it to relax at a higher concentration of calcium. So we're throwing more calcium out there, we're pulling it back, and it's relaxing faster. So you've got these two targets that are important for increasing the relaxation rate. Excuse me. And then kind of as a sideline here, we also phosphorylate protein phosphatate inhibitor. So PPI will block the phosphatases in the cell. So protein kinase A goes out and phosphorylates a bunch of proteins, and that phosphatase is always there taking those phosphates off. And by activating this PPI, you're slowing down the phosphatases, so you're potentiating the effect of the protein kinase A. Okay? Does that make sense? Okay. Wow. See, half an hour I got through all of this. Normally I would take a whole class to do this, but I have an animation here I want you to watch. Sometimes when I do these Facebook animation or YouTube animations, they don't play so well. It's here in the PowerPoint, so if you can't see it very well, you can come back to the PowerPoint later and watch it. But this is about a 6-8 minute video. It's talking about inotropy. So inotropy is about the strength of the contraction. A positive inotropic agent would make it go faster. So a beta agonist would be a positive inotrope. They'll talk about digoxin or digitalis or something like that. A couple different drugs like that. They're going to affect the sodium calcium exchanger. So I warned you to watch that. Okay, let's see what we got here. Let's do a new share. Here it is.
Inotropes are medicines that alter the force of contraction of the heart. By definition, there are positive inotropes that strengthen cardiac contraction and negative inotropes that weaken it. However, when not specified otherwise, the term inotrope usually refers to positive inotropes, which are the topic of this video. Negative inotropes, such as beta blockers and calcium channel blockers, significantly overlap with antiarrhythmic agents and are covered in another video. Inotropes are used in conditions where there is a sudden low cardiac output, such as acute heart failure or cardiogenic shock. Cardiac output is the product of stroke volume, the amount of blood pumped in one heartbeat, and heart rate, the number of beats in 1 minute. More forceful contractions induced by inotropes pump more blood out of the heart, increasing stroke volume. Many inotropes also accelerate heart rate, which contributes to increased cardiac output. Cardiac muscle contractions are triggered by a surge in intracellular calcium concentration. When cardiac muscle cells are stimulated by action potentials, calcium channels open, allowing calcium to rush in. This calcium influx activates ryanodine receptor on the membrane of the sarcoplasmic reticulum, the SR, causing more calcium release from the SR in a process known as calcium-induced calcium release. Calcium then binds to troponin units on actin myofilaments and sets off muscle contraction by the sliding filament mechanism. Most inotropes act by raising the levels of intracellular calcium. They can also be called calcitropes. Many calcitropes operate via the catecholamine pathway. They bind to beta-adrenergic receptor, activating a signaling cascade that leads to production of cyclic AMP, CAMP, a second messenger. CAMP promotes phosphorylation and hence activation of both L-type calcium channel that allows calcium influx from the extracellular fluid, and ryanodine receptor that mediates calcium release from the SR. The result is an increase in intracellular calcium that drives a stronger contraction. Most catecholamines have a short half-life and are given by continuous infusion. Some other agents are inhibitors of the enzyme phosphodiesterase 3, PDE3. Because PDE3 breaks down CAMP, inhibition of PDE3 results in accumulation of CAMP and subsequent increase in intracellular calcium. Digoxin, a widely used inotrope, acts by inhibiting the sodium-potassium pump, causing intracellular sodium concentration to rise. This then leads to higher levels of intracellular calcium via the action of sodium-calcium exchanger. The problem with most calcitropes is that they increase contractility at the expense of cellular energy, meaning the heart muscle requires more oxygen to operate. This may lead to adverse effects, including higher mortality. Other strategies have been explored to design inotropes that can boost cardiac output without increasing myocardial oxygen demand. Levosimendan is a calcium sensitizing drug. It enhances troponin C sensitivity to intracellular calcium, thereby increasing the force of contraction without consuming more cellular energy. Omecamtiv mecarbil, OM, is a new drug that is still under investigation. OM increases the efficiency of heart muscle contraction. It binds to cardiac myosin, stabilizing myosin-actin connection. This increases the number of myosin heads bound to actin, producing more force during cardiac contraction.
Okay. So I think that's pretty good. It kind of explains some of the things I talked about. Threw a couple of drugs in there. Honestly, I didn't know about those last two drugs, but it talked about digoxin. Let's talk about that real quick, and then we'll talk about the other two. Okay, so talked about what we talked about with FKB 12.6. They didn't name it, but they said phosphorylation of that. They also talked about phosphorylation of the calcium channels. So over here, your NCX then is controlled by the concentration of sodium. So when you change the sodium gradient across the cell, that changes the calcium calcium movement. So digoxin will block sodium. It will block the sodium potassium ATPase, which then will decrease the amount of sodium coming in, which decreases the amount of calcium going out. And as a result, you get more calcium loading in the SR and you've got more activator calcium. So a contraction with digoxin in there, okay, this is your unstimulated. With digoxin, the time course would be the same, but it would be a stronger contraction. Okay, so it's a positive inotropic agent. So it's one of the few drugs available that will make the heart beat stronger. Usually if a heart's in failure, the whole cyclic AMP system is so overused by the heart that you can't get much more out of it. But this will make the heart beat stronger. One of the problems with digoxin, though, is called digitoxin for a good reason. If you remember cell biology, the sodium potassium ATPase is in all your cells, and it's extremely important in action potentials, right? Controlling the sodium and potassium concentrations. So if you go to too high concentration of digoxin, you end up getting arrhythmias in the heart, and then you die anyway. So it's a dangerous drug. It's, you know, when I was in grad school back in the 80s, they said if it was a new drug coming on the market today, it would never make it on the market because the difference between a therapeutic dose and a toxic dose is so narrow that you have to be really careful with that drug. It's been around for like ever, basically. There was a report written in like the 18th century of some guy who used the plant foxglove to make people's to help for congestive heart failure. Back then they called it dropsy because you tended to fall. If you've got congestive heart failure and you try to exercise too much, your heart can't keep up and you fall over. And you've got like fluid buildup in your legs and everything. So they were using this plant, foxglove, which has got digitalis in it, to increase congestive heart failure. So they've been using this drug since, you know, at least the 18th century, but probably like since there were foxglove plants around. I don't know. But anyway, that's digitalis, positive inotrope, and it works through the NCX. So if you inhibit NCX, more calcium stays in the cytoplasm and more calcium gets loaded in the SR, more activator calcium. The other ones she talked about, I don't have a picture of, no, I don't. Okay, so the other thing she talked about was a calcium sensitizing agent, levisimensidone or something, I forget exactly, but it binds to the troponin C and makes it bind calcium better. Okay, so you need less activator calcium to get a stronger contraction. And the other one was a binding to the myosin head and causing it to be more efficient in how it grabs the actin. So 2 new positive inotropic agents that I didn't know about. We all have something to learn here. Okay. Okay. All right, let me open it up to the crowd for questions, and then we're gonna move on. Any questions? No, Dante says no. Okay, Dante, maybe I have a question for you. No, I'm just kidding. Okay, so that is contraction of the heart muscle and how it's stimulated by beta agonists. So let's talk, this class, I always do a lot of original research paper reading because I think it's important once you get into 400 level classes to learn how to read these papers. Now, papers in our field, in cellular biology and stuff like that, are always so much more complicated than, like, you know, environmental science. I can read an environmental science paper and make sense out of it. These things, there's so much embedded in them, it's hard to really understand them unless you've got the background for it. So I want to start out by, before I just tell you to read this, explain some of it so that it can make more sense to you. So here's the paper, and this paper is in your Moodle page in a PDF, so open it up. One thing about reading these papers, I would say start out reading the introduction. That's really important. And then skip over methods. Don't even look at methods unless you need to understand something that doesn't make sense from the figures. So introduction, then the results, discussion, and then go back and read the abstract afterward and see if it all makes sense to you. So read it in that order if possible. And try and get out of the habit of just glossing over words you don't know. I know I had always done that before when I was your age. There's a word I don't know, well, I don't know, maybe it'll make sense later. Just keep reading and reading. You ever read something and after a while you realize that your eyes are on the end of the paragraph and you don't know what happened between the beginning and the end because your mind was somewhere else? That's the problem with reading these papers. It's really hard. So you really got to focus on this to understand it. But a couple of things you should know. Going in. Hemodynamics. This is something that that video talked about a little bit. So they're going to talk about, the paper is about when somebody gets an infection and it goes septic. Septic means the the bacteria got into the blood, so your whole bloodstream is now septic. What happens in sepsis is your cardiac output goes up for a while. Okay, your heart beats faster and stronger, and then it crashes and you die. So they've got this rat model where they open it up, take the intestine and cut it off and open it up. So all the bacterial and stuff goes into the peritoneal cavity, and they give the rat a pretty repeatable infection that way. So it's a good model. It's not good for the rats, frankly, but it's a good model for infection because it's very repeatable. So cecal ligation and puncture, they call it. They ligate at one end of the large intestine, and then they puncture it, and they let the bacteria come out, and they get a very repeatable infection. So when they do this, they see cardiac dynamics go up and then down and then death. So cardiac output then is the amount of blood ejected from the heart per minute. And it's a function of the stroke volume, how much comes out per beat, and the number of beats per minute. So for a human, typically it's about 80 mils per beat, 70 beats per minute, about 5.6 liters per minute. That's quite a bit. If you take your aorta out and hold it, it will hit the ceiling and higher, and you'll be dead in like probably 10 seconds. Okay, because all your blood will come out all at once. I think you've only got 5 liters, so okay. That's neither here nor there. All right, so the important thing for us, though, is stroke volume. Stroke volume is all about inotropy. So inotropy is the strength of the contraction. Positive inotropy increases the stroke volume. So the stronger you contract, the more of that blood that's in the heart will actually come out. And when we get into the hemodynamics in about a month, we'll talk about stroke volume and how stroke volume is controlled. But for now, just think contract stronger, more blood comes out. And your voltage-dependent calcium channels will play a big role in this, right? They're the trigger calcium. More trigger calcium gets more activator calcium, stronger contraction. So VDCCs, another thing you need to know about the VDCCs for this paper is that there are drugs called dihydropyridines. Dihydropyridines are calcium channel blockers. Now these will bind to the voltage-dependent calcium channel in the heart and block it and cause the heart to stop beating. That's not what they're used for clinically. Clinically, they affect arterial muscle more than they do cardiac muscle. And so they relax veins and they decrease blood pressure. But they're also good research tools for looking at the voltage-dependent calcium channels in heart tissue. So in this study, they're going to be using dihydropyridines as a marker to find how many the voltage-dependent calcium channels. I'm about to die here. Hang on a second. Okay. I don't know why. All right, I better shut up and get it done, get over with. Okay, so voltage-dependent calcium channels will also be called dihydropyridine receptors. So in this paper, when you see dihydropyridine receptor, they're talking about the VDCCs. All right, now their binding studies, you know, I showed you semi-log plots where you've got like the number here and then the log over here. They don't do that. They only show the straight up numbers. So these are your binding curves. Now, what's important for this is Bmax. Where is the Bmax in each different fraction? And I'll talk about fractions in a second. Settle down. And right next to these binding curves, you'll see Scatcherd plots. You can ignore the Scatcherd plots. They're basically a linear transformation of that binding isotherm. So instead of doing a non-linearly squares curve fitting, you're converting it into a linear thing. And then the slope is the KD and the Y or the x-intercept is the Bmax. You can ignore this if you want, or you can just say, okay, all I care about is the Bmax. Where's the Bmax? This is low and this is high. What else? What did I just talk about? Oh, the different fractions. So, so basically what they're doing in this study is they're looking at sarcolemmal dihydropyridines, which are from the cell surface, versus light fraction dihydropyridine receptors, which are calcium channels that are in this subcellular vesicle fraction. So when they say fraction, basically they're taking heart tissue, grinding it up and doing differential centrifugation, and they're looking at things that come out in big vesicles versus big things that come out in small vesicles. So this light fraction are these dihydropyridines that are right below the sarcolemma. And the cell can regulate how many calcium channels are in the surface by bringing these up and putting them in the surface, or bringing them down and taking them out of the surface. So what this sepsis is going to do is put these dihydropyridine receptors up into the surface. At first, you get a stronger contraction, and then later pull them down out of the surface, and you get a weaker contraction. So that's the hypothesis here. That's what they're expecting to see. So you've got control, and then you've got sarcolemma, and then you've got light fraction. Keep an eye on that. And another technique that they're using is photoaffinity labeling. So with this, oh, look at the 2 L's there. That's only 1 L. So with this, basically, you've got a whole bunch of proteins in your prep. I know. And you're binding the drug to the receptor you want. Oh,